just be online. He will present. He will start presentation. He will soon resume his presentation. Please just be online. ಏನಾಯ್ತು ಅವ್ರಿಗೆ? Yes, sir.
That is whatever the nano particles we have put.
Sir, you start your screen, sir. You start your screen, sir. Yeah, I think you have just logged out, I guess. ಮತ್ತೆ ಇನ್ನೊಂದ್ಸತಿ ಸ್ಕ್ರೀನ್ ಶೇರ್ ಮಾಡಿ ಸರ್.
Okay.
ನೀವು ಇನ್ನೊಂದ್ಸತಿ ಸ್ಕ್ರೀನ್ ಶೇರ್ ಮಾಡ್ಬೇಕು. ನೀವು ಐ ಥಿಂಕ್ ನೆಟ್ವರ್ಕ್ ಪ್ರಾಬ್ಲಮ್ ಇತ್ತು ಅನ್ಸುತ್ತಲ್ಲ.
Now?
Yeah, we are able to see. We are able to see.
Okay.
Yeah.
The first one, what I was talking about, the electromagnetic enhancement associated with the roughened metal surface. I told you the substrate, whatever the substrate we are going to use for putting the sample, is now added with silver and silver and gold nanoparticles, so that
sir again ಮತ್ತೆ ಇಲ್ಲಿ ಸ್ವಲ್ಪ ಪ್ರಾಬ್ಲಮ್ ಮಾಡಿದ್ರು ಸರ್. ಮತ್ತೆ ಶೇರ್ ಮಾಡಿ ಸರ್. Sorry sir. Sorry for disturbing you.
No problem. Tell me what is the problem. We will solve it.
Yeah. Just reshare it. That's it.
You are presenting to
now
everyone.
we are able to
is
see.
Is that right now?
Start.
Okay.
Hello. Elidra sir. Elidra.
There is an enhancement of this one of the electromagnetic enhancement, which is associated with the roughed metal surface, because the silver and gold particles are put. The other one is called as the chemical enhancement due to the electronic coupling of the molecules adsorbed on the roughened metal surface, which means that there is a sort of bonding which gets formed, and the analyte will be we will be able to analyze the analyte much more easily. The electromagnetic effect is supposed to be more dominant than the chemical effect. So let us see what is this electromagnetic enhancement. The one is that we have the concept of surface plasma, which means that if the metals like Ag and silver, which are very small in this one, if they are located, then when the electromagnetic wave falls on them, they create plasmons, and these plasmons oscillate perpendicular to the surface of the metal, and hence causes better scattering. Causes scattering, reflection, and the roughness of the surface, which can either the physical roughness or maybe produced because of the air particles. Such a thing will enhance the electromagnetic resistance because of the surface plasma. Hence, the signal to noise ratio increases quite a lot. Hence, the specially prepared metal surfaces like gold, silver, and copper, you can see the Raman signal will get enhanced by 10 to the power 4 to 10 to the power 6 fold, which enables faster and higher accuracy of detection. And hence, this can be used for biological and chemical samples. And again, as the signals obtained by normal Raman scattering is usually very weak, so in order to get more detectable weaknesses, one prefers this SARS technique, surface enhanced Raman spectroscopy technique. This technique can yield information how molecules interact with surfaces, which allow the detection of very low concentration of analytes. And it has been used successfully on nickel and platinum electrodes having different surface roughness, and helps in obtaining good quality surface Raman signals from transition metals also. The Raman spectroscopy will be further developed as a versatile mean for characterizing the interfacial processor, where the roughness and electrochemical and other surface environments, how they affect is being still looked at. Quantitative analysis of fungicide theorem has been done using silver nanoparticles. How they make the silver nanoparticles, probably Dr. Manjunath will be able to discuss with you. They do what is in an aqueous solution, they do the radiolysis and produce the silver particles, and they don't add any of the aggregating agent, so that the nanoparticles get capped. Here we have that, so that you will have m size of the Ag particles, which will limit the impurity part. Hence, they will be able to give a better SARS signals. Similarly, metallite green isocyanate has also been observed on gold particles, and the signal is known to increase by 20-fold time. And Raman spectrum is also described to yield using gold nanoparticles with ultra thin silica or alumina as the substrate. and a monolayer of such nanoparticles is fed over the surface, and hence probed, and then the analyte is put on such a surface, and the Raman spectra is probed. So this supports the application of SERS in materials, life sciences, as well as in the field of food safety, drugs, explosives, and environment pollutants. So you can see the one which was such an effect, what you had seen in 1930, today you can see with nanogram of the material, you will be able to do the analysis. So it also gives flexibility to analyze hazardous environmental samples, because you need not have to inhale or anything like that. So detection of methylene trinitramine, it is called as RDX. You would have heard of this RDX. It is called as Royal Explosive Device. And there are many methods people are trying to see RDX, because that is being now transported in every airport, and then you will see some bomb blast and other things. So in order to detect that, people are using now SCRS method, and it is done by putting gold nanoparticles 90 to 100 nanometer in diameter, and the detection level of 0.15 milligrams per liter can be seen from contaminated groundwater sample. Hence, SCRS technique, you can see how very good, how it is so nice in detecting the analyte. Similarly, one can detect TNT, which is also another explosive which is used by people for damage, perchlorate, perrenate, and uranium in groundwater. All such things can be used, can be found out using SCRS technique. So hence, SCRS technique can be used to distinguish explosives in the solution using azote dyes. If you use azote dyes, then there will be a coupling of azote dyes with this, and such a complexation will make the molecule very nice for SCRS detection. Hence, the detection up to nanomolar. You can see nanomolar, 10 to the power of minus 9 moles, you will be able to detect. That is one of the avatars of Raman. I will be touching on the second avatar, which is called as the confocal Raman microscopy. This is invented in 1955 by Marvin Minsky, and see, this he used to see, people use it to study brain tissues by slicing them. Whereas using the confocal Raman microscopy, one can use this entire probe head, which can be made into the microscope objective, and it is scanned in such a way that you will get a three-dimensional image of that. So the real slicing of brain tissues can be avoided by using this microscopy. So I am just telling you now how Raman is being used for different purposes, depending upon how changes have been made on the Raman. So here, the filtered Raman signal then returns to the back to the spectrometer. I will show the black diagram. You will be able to understand it better, where it is dispersed on a CCD camera and to produce the spectrum. So, but fluorescence background should be sufficiently avoided. For that, people put sufficient filters. Okay,
Sir,
and
ಮತ್ತೆ ಇನ್ನೊಂದ್ಸತಿ ಮಾಡ್ರಿ ಸರ್.
ಇನ್ನೊಂದ್ಸತಿ ಮಾಡ್ಬೇಕಾ?
ಪ್ರೆಸೆಂಟ್ ಮಾತ್ರ.
ಓಕೆ,
ಇಲ್ಲಿ ಹುಡುಗರು ಅದು ಗೊತ್ತಾಗಲ್ಲ ಸರ್ ಅವ್ರಿಗೆ ಅದು.
ಕರೆಕ್ಟ್.
ಹಾ ಸರ್.
Okay. So one can get a three-dimensional image through the turbid medium, high spatial resolution, and rapid identification of micrometer-sized biological specimens. Now you see, Raman was used by chemists. I know the physicists used. I know. Now I am saying biological specimens are being seen using the confocal Raman microscope. This is the typical block diagram. You see, you have a laser which is now put onto a beam splitter, which is a dichromatic mirror, and then it goes to the microscopic objective, which means that now it is focused in such a way that even if I have micrograms of the sample, then that is good enough that it can be focused, since I have the microscopic objective. Then once the scattered radiation